So thank you for inviting me to just give a bit of information of the research I've been conducting over the last year. Looking at the role of glucagon signalling in MND. So there have been various studies that have shown a link between diabetes and MND, suggesting that there's an increased risk of MND in people with diabetes. And there is some evidence that suggests that diabetic symptoms may negatively impact disease progression. So diabetes is basically an inability, is there the laser? Sorry. There we go. So the inability to maintain blood glucose levels. So after eating, when we have high glucose, the pancreas releases insulin, which drives then glucose uptake into various tissues to normalize blood glucose levels. Whereas in periods of fasting, such as like overnight, when we have low glucose levels, the pancreas actually releases glucagon, which acts primarily upon the liver to release our glucose stores, which are stored as glycogen, to then restore blood glucose levels. And so a lot of the research looking at diabetes and MND have focused on this side of the pathway and looking at insulin signaling, whereas we were actually interested in looking at the opposite effects of glucagon signaling. So along with maintaining blood glucose levels, glucagon signaling can also be involved in the liver to break down glycogen to produce glucose, but it's also involved in fat storage within the liver. It also increases fat storage and fat mass in adipose tissue and can be involved in the secretion of insulin. So glucagon can actually also play a role in that insulin signaling side of glucose homeostasis as well. And so for my research, I've focused on 2 mouse models of MND. So SOD1 G93A and TDP43 are known mutations to cause familial MND. And so we use 2 transgenic models that overexpress. So first is mutant human SOD1 with the G93A mutation. And we also have used the TDP43 model, where there is an overexpression of mutant human TDP43 with the Q331K mutation. And just to show with the SOD1 mice that over their lifespan, we see a significant drop in muscle strength, and also a loss of body weight towards the later stages of the disease progression, which ultimately leads to reduced survival in these mice. And so the TDP43 model also sees symptoms similar to this, however, it's a longer profile. And for the data that I'm going to present today, we look at approximately mid-symptomatic stage of both these models. And so, as I mentioned, that glucagon signaling is involved in maintaining blood glucose levels. So if we see an impairment in glucagon signaling, we would expect that fasting blood glucose levels to be reduced. And indeed, when we fasted these mice for 6 hours, which would be equivalent to fasting overnight for humans, we see here in the blue, in the SOD1 G93A mice, that there is a reduction in blood glucose levels, and we also see the same in the TDP43 mice, so suggesting to us that there may be impairments in glucagon signaling. And to confirm this, we ran a glucagon challenge, which is similar to a glucose tolerance test that people will take when they're pregnant or if you're suspected of having diabetes, except in this case we actually inject glucagon into the mice and then we measure their blood glucose levels over time with the expectation that the glucagon will raise blood glucose levels. And what we see in the SOD mice is that here in the blue are the SODs. When we inject them with glucagon, there is a lower response to glucagon, and it raises the blood glucose levels to a lesser degree than our wild-type mice. And we can see that by just looking at the area under the curve, which is basically this area underneath the response, so this area down here. And again, in the TDP-43, it's less pronounced, but we also see that this is reduced. So this is suggesting or further confirming to us that we do see reduction in glucagon signaling in both these mouse models. But as I mentioned, there are other symptoms that can indicate impairments in glucagon signaling, such as in the liver, if we have impaired glucagon signaling, we would expect glucose storage as glycogen to increase, as well as an increase in fat storage. And indeed, we see an increase in both the SOD1 and the TDP-43 mice in their levels of glucagon, which shows, of glycogen, which means that instead of using glucose, these mice are actually just storing the glucose as glycogen in the liver. When we look at their fat levels, we also see an increase in both the SOD1 and the TDP-43 mice. So again, this indicates that we do see these impairments in glucagon signaling. Finally, we looked at insulin secretion, and as I mentioned before, if we have impairments in glucagon signaling, we would also expect the insulin secretion from the pancreas to be reduced. And so here we performed a glucose tolerance test and collected the blood over different time points, and then used this blood to measure insulin levels. And as you can see here for the SODs in the blue, that in response to glucose, we see a marked decrease in insulin release, whereas in the wild types, we see the expected increase of insulin levels in response to glucose. So overall, what I've found is that, so normal glucagon signaling, we would see an increase in glycogen breakdown and fat storage in the liver, increase in fasting blood glucose levels, an increase in insulin secretion, and also an increase in fat mass. But if we actually look at the metabolic dysfunction that I've shown you today, but also that has been reported in patients, we see that in MND, there's a decrease in fasting blood glucose levels, a decrease in glycogen breakdown, leading to increased glycogen stores in the liver, an increase in fat storage in the liver, as well as a decrease in insulin secretion. And although I haven't shown the data here today, there is evidence in both patients and mice that there is a decrease in fat mass in MND. So overall, this has led us to want to treat the SOD1 mice with a drug that can stimulate glucagon signaling. So it's called IUB-288, and basically it binds to the glucagon receptor and stimulates signaling, and it stimulates signaling to a greater extent than glucagon itself. And so we'll be treating these mice 3 times a week and looking at the parameters such as the grip strength and survival to see if it can improve these parameters in the mice, but as well as looking at other metabolic phenotypes, as I presented earlier, to see if it can change insulin secretion and the liver storage of both fat and glycogen as well. And hopefully in the coming months, we will find some data that is positive to show that glucagon treatment is a good target to treat MND. But overall, why is glycogen important in MND? I guess this is the take-home message that everyone wants to know. Well, glucagon signaling maintains blood glucose levels, and this is vital as glucose is energy for all organs, including our muscles and the brain. Glucose provides the energy for every single function that we do, every movement of muscle, every function of our neurons requires glucose. So if we don't have access to these energy stores and instead this glucose is being stored as glycogen in the liver, then we're actually losing access for these cells to this energy, and it may actually increase the vulnerability of these cells to insults. So although we don't think that glucagon may be acting on neurons directly, this loss of energy can actually potentially increase cell death just by limiting their energy sources and their ability to actually deal with these insults. And so we're hoping that improving glucagon signaling may be able to slow disease progression and potentially improve quality of life for people with MND. But with that, I'd just like to acknowledge Dr. John Lee and Professor Trent Woodruff of the Neuroinformation Lab, as well as in particular the Fat Rabbit MND research grant that has allowed me to actually continue this research this year. Thank you.